Jonathan is actually an MD, and I want to tell this little anecdote because I find it very beautiful. And he was set to become a medical doctor. He was interested in oncology, but with a certain focus on the liquid part of oncology. And then at night, when he was working too long, as he used to do sometimes, he saw this poster talking about something weird. He was not knowing what it is. It was called oncogenes. The person supposed to talk was Bob Weinberg. And when attending this seminar and learning that you can actually take a gene and put it into mouse cells and then into a mouse, and that grows a cancer, and this gene is only one amino acid different from the normal gene, and it's called RAS. That is important and meaningful, and this turned him into a molecular biologist. And he then moved on to become a postdoc at Dana-Farber at the lab of Ulla Hansen, which again is progeny from the oncogene field because she was a former Phil Sharp lab member. And this really turned him into becoming a molecular biologist, but always thinking from the side of cancer and liquid cancer. And even already in this lab, he already became interested in transcription regulation and in extension then later to chromatin epigenetics. And of course, you know, many studies of the last 20 plus years where he has shown beautiful examples of how this knowledge can actually be useful to understand what's happening in the clinic. And he's now director of one of the most important cancer institutes, and you know him from the Florida University, and it's a huge pleasure to have him here. John, the audience.
Thank you very much, those of you here and those of you at home or online. Thank you very much for being here today. What a pleasure to be here with you and meet so many interesting scientists already today. I'm looking forward to the rest of my visit as well. So welcome to University of Florida. We are located here. Miami is here, Tampa is there. So people come to Florida, a lot of times they go to Miami Beach. We're way up here. This home of these springs at 70 degree, well, that's Fahrenheit. Don't ask me for the Celsius, okay? Something nice, warm enough. Winter and summer, there are these springs of perfectly clear water, like 70 feet of water. It looks like it's 2 inches, like the pool outside because it's so fresh. And there are manatees in here, these sea cows. Sometimes there's alligators in there. People go canoeing there. And the other big thing in Florida is that this is outside the football stadium. These are our football heroes, and there's also alligators out there too. But so welcome from the University of Florida. I'm going to talk about today one particular histone methyltransferase that's affected in a number of cancers, but it was discovered and brought to the attention to the cancer field in multiple myeloma initially. And it's a protein that's got a few different names: MMSEP for multiple myeloma set domain protein, WHSC1 for Wolff-Hirschhorn syndrome candidate gene number 1. And actually, Kurt Hirschhorn was a colleague of mine at Mount Sinai, so it's nice to meet the person who's got a gene name for them. And it's more recently been called NSD2, which is a lot more boring, but it's a lot more scientifically, standardized. So this is one of about a half dozen genes recurrently overexpressed by virtue of aberrant immunoglobulin gene rearrangements in multiple myeloma, which is a tumor of very late B cells that are really immunoglobulin factories. And this was traditionally a poor prognostic feature, though more modern therapy has made this a little less worrisome. But this is where it was initially discovered, and it is one of a number of these histone methyltransferases in the NSD2 family that particularly catalyzes the histone 3 lysine 36 dimethyl mark, which is a gene activation mark, a mark that precludes gene inactivation. And it's also been found NSD2 tends to be very highly upregulated in many metastatic cancers, metastatic prostate cancer. It's actually quite a good biomarker of an effector of metastases in prostate cancer. It has a number of chromatin binding domains of different names, PWWP, PhD. Some of these have had crystal structures shown and are shown to recognize specific chromatin marks. and critically has this histomethyltransferase domain in the set domain, which every histomethyltransferase except one, which is .1L, have this set domain, and it's a canonical domain that takes acetyl groups and takes methyl groups, places it on chromatin. Okay, so this is where we started with in multiple myeloma, and one thing we did is we got a hold of all of these T414 myeloma cell lines that had this overexpression of NSD2. Notice that this is the full length. There are two isoforms, and then there's these truncations here that occurs because the chromosomal breakpoint occurs sometimes in the intron that encodes between the two exons that encode this PWWP domain. So we inferred that this domain, which actually is important for very tight chromatin binding, is not really necessary for the potential oncogenesis, because all these cells really disrupt this domain, but they still have NSD2 overexpression. And what we found critically in these studies about a decade ago is there's a huge shift in the K36 dimethylation is increased in all these cell lines relative to other myeloma cell lines, and there's a reciprocal decrease in K27 trimethylation. And this, summarizing some mass spectrometry work we've done with Neil Kelleher, it's known that the major catalysts of K36 dimethylation are these NSD2 family, though there's a few other enzymes that can do it. The other major modifications that methylation of histone is K27 di and trimethylation catalyzed by EZH2. These two actions are completely antithetical to each other. There is no such thing as a histone 3 lysine 27 trimethylation in the histone tail and a K36 dimethylation. You can't detect it by mass spectrometry. It just doesn't exist. So these two systems are set up and diametrically oppose each other. K36 dimethylation tends to be on genes that are activated. K27 trimethylation tends to be on genes that are repressed. And when we did global ChIP-seq studies in our myeloma cells of two types, one that had high NSD2 and an isogenic pair, which is NSD2 low, we found that the K36 dimethyl levels drop, the K27 trimethylation levels rise. But what happens at the ChIP-seq level? In the NSD2 high cells, you see this K36 dimethyl mark basically everywhere. Gene promoters, bodies, enhancers, it's just everywhere. Whereas when NSD2 is low, you get more of this centering around, sometimes around promoter sites. So you have this really wipeout of the normal architecture. And another thing that you find here is that if you take a look at do ChIP-seq for K27 trimethylation, you find genes that are repressed in NSD2 low cells, and this K27 mark is gone in the NSD2 high cells. So when you have NSD2 high, you have the K36 dimethylation, you get a big decrease in K27 trimethylation pretty much globally, and you get an aberrant gene activation pathway. Now, Jane Skok at NYU has done this as well, and she found additionally that there is, when you have K36 dimethylation is high and NSD2 high cells seen in red, there's a K27 is low. She also finds new K27 acetylation peaks and some new attack-seq peaks as well, suggesting that globally, you're getting enhancer activation when K36 dimethylation is present with NSD2 is high. And what you seem to be doing is you're keeping the EZH2 polycomb complex away from chromatin and keeping genes in a potentially activated state. Other work is also shown from particularly Keiko Zato at the NIH that NSD2 is involved at the transcription start site to binding to PTEF-B and BRD4 to help the initiating RNA polymerase go into an elongation state and helping deposit the transcriptionally coupled histone 3.3 in these initiating and elongating chromatin. And similarly, they've also found a handoff of NSD2 to the intragenic histone methyltransferase known as SETD2, which creates a trimethylation mark within gene bodies that prevents additional rounds of transcription. Collectively, though, all this keeps this EZH2 at bay and keeps it off the chromatin and away from repressing genes. So we could just think about, globally for today, we could think about NSD2 as something that defeats the repressive polycomb complex. And we'll see how this comes into play in my major discussion today, which is, because this is the Leukemia Institute, we're going to talk about NSD2 in acute lymphoblastic leukemia. Now, with modern genomic and exomic sequencing, pretty much we're reaching the end of the discovery phase of what genes are mutated in cancer, and we know that epigenetic genes encoding epigenetic regulators amongst the most common in all cancers. And as seen here in this study, the WHSC1, it's NSD2, is frequently found across many different subtypes of childhood ALL as being recurrently mutated. And another study showed that it's particularly found to be recurrently mutated in very early relapse or early relapse of acute lymphoblastic leukemia. And there are patients that will have 1% of their cells that will have an NSD2 mutation at diagnosis and 100% a relapse. So this mutation seems to be enriched as progression occurs in these patients. And these mutations, though, it is a specific point mutation in the SET domain, usually at this residue known as E1099K, but more occasionally at this residue T1150A. And the mutation particularly is found also in mantle cell lymphoma, but in acute lymphoblastic leukemia, most of the mutations are at this single residue. So the way I came upon this mutation was when the Novartis Cell Line Encyclopedia came online. I was at AACR, and Bill Sellers, the former head of research at Novartis, was telling me backstage at one of these presentations, oh, we got this new database of every cell line. We've done genome sequencing and all the RNA sequencing on these cell lines. So I went back and typed in my favorite gene, and out I found all these point mutations at this single amino acid. I said, well, these have to be gain-of-function mutations. And that's how it actually happened a little bit behind the scenes. So we actually got a hold of these NSD2 mutant ALL cell lines that we identified by searching the Broad Cell Line Index and the Novartis Broad Cell Line Index, and we found that these had increased K36 dimethylation and decreased K27 trimethylation, just like we found in multiple myeloma. Same thing, a little bit quantitatively different. This difference by mass spectrometry is about a 3-fold difference, whereas in myeloma, it's about a 7 to 8-fold difference. So, but there's still a big switch globally of the chromatin. And this mutation doesn't occur in the enzymatic domain where S-adenosylmethionine binds in the catalytic site. Instead, it occurs at the entrance to the site where the enzyme is just touching chromatin. And with a computational biology colleague, we modeled this, and we actually posited that there's increased binding area and increased free energy of binding of NSD2 to chromatin with this extra amino acid change. And we could actually predict the amount of surface area binding, and it seemed that it was particularly avid at binding the intergenic histone 3.1 rather than 3.3, which is found in transcription sites. So this NSD2 would bind well to histones that are replication-dependent histones that are found all around the genome. We also did Michaelis-Menten studies, and we found that the K-cat of this NSD2 1099K was 3 times that of wild type, and this 1150A is 5 times that of wild type. So this mutant enzyme, gain-of-function enzyme, pretty unusual. Most epigenetic gene mutations in cancer are loss of function, very few gain of function. This mutation, point mutation, opens up the enzyme to bind chromatin, has an increased turnover rate, And more recent structural studies, actually elegant studies of NSD1 and NSD2 kind of show the enzyme, how it interacts with chromatin. And one more recent paper suggested there's an auto-inhibitory loop that inhibits the enzyme's activity. These point mutations affect that auto-inhibitory loop. So for multiple mechanisms, perhaps increased chromatin avidity, getting rid of a autoinhibitory loop, this is a more active enzyme. It doesn't change its substrate specificity, but it changes the rate at which it converts an unmethylated histone to a dimethylated histone. Now, in terms of the avidity for chromatin, we can actually measure that in vivo using a FRAP assay, where we took GFP labeled NSD2 and transfected into cell, and then burned a little hole with a laser and measured the fluorescence recovery time. And we find that this is the recovery of wild type NSD2. The point mutant NSD2 has got a slower recovery, meaning once it's bound to chromatin, it doesn't come off chromatin as avidly. It goes there like a dog with a bone and it's chewing on that chromatin and putting on that K36 dimethylation mark. So a lot of evidence that this is a functionally gain of function mutation. But the proof of the pudding here was actually editing out this mutation. There are about a half dozen acute lymphoblastic cell lines commonly used in many laboratories, including one I saw today from one group, SEM cells. I think Alejandro was using that cell that has an NSD2 point mutation, and it has high levels of K36 dimethylation. If by CRISPR gene editing, either converting that back to wild type or removing the mutant allele, we lose that K36 dimethylation and we gain back the K27 trimethylation, showing functionally, proving in these cells that this indeed is creating this changing chromatin environment. And we've actually done what is sort of a virtual rate studies using heavy and light labeling of cells with heavy and light isotopes, so we can calculate the virtual rate constant of NSD2 in wild type cells and mutant cells. And we find this rate here is of about 4, is the rate at which unmethylated K36 goes to monomethylated, whereas in the 1099K cells, there's the rate is about 4-fold higher at nearly 14. So it means the initial encounter of this enzyme with unmethylated histone, that methylation goes much faster. The second step of mono to di, not that much faster, but that initial step much faster. So the initial attack of this enzyme on chromatin, it binds better, it sticks there, it turns over and creates methylations more quickly. So what I can say here is this gain of function mutation of NSD2 increases activity, increases interactions with histone, blocks autoinhibition, and increases K36 dimethylation levels in acute lymphoblastic leukemia cells in these children. We've not found it in adult ALL, we've only found it in childhood ALL, both T and B cell. So what happens if you remove this point mutation? What we found is that the cells grow much more poorly, seen in blue. One of these cells we could barely keep in culture. One of the cell strains we actually lost. We have another one we were able to maintain, but they have decreased growth. The other thing we found is that there's increased apoptosis rate in these cells in blue where we removed the point mutation. So there's something anti-apoptotic about this mutation in this ALL cell line. But it also creates other functional properties comparing the NSD2 mutant versus wild type cells. Here we're trying to say how sticky are these cells to a cell layer. In this case, we're actually seeing how well they stick to meningeal cells. And the mutant cells are in green and the wild type cells are in red. And we see the mutant cells stick better to a meningeal cell layer here. So they're stickier. And this will why meningeal cells will become apparent in just a moment. We've then functionally tested these isogenic cell pairs in xenograft animals, injecting wild type or the mutant cells into these mice. And what we found is that the NSD2 mutant mice needed to be sacrificed due to terminal disease at a much faster rate than the NSD2 wild type cells in these mice. And we also measured the photon flux of these luciferase tract cells. Now, this was kind of curious because these NSD2 wild type mice could have considerably more. Take a look at the scale here, 10 to the 10th, 10 to the 11th, nearly 10 to the 12th. 10 to 50 times more leukemia was tolerated in these mice if the NSD2 mutation was removed. So there's not just, there's something qualitatively different about the disease with NSD2 mutation. And what it was is that these NSD2 mutant cells were homing into the mouse brain at a much different rate than the wild type cells, where you could find it in the brain, but in the rest of the body kind of homogeneously. So these cells became neurotrophic, and we could quantify this in a simple ratio of head to body photon flux. There's about a 3-4-fold increase in the relative number of cells going into the mouse nervous system. So what we actually found is normally acute lymphoblastic leukemia cells, if they go to mouse brain, line the meninges seen here. But what we actually found is these mutant cells, is not only did they line the meninges, but they invaded the mouse brain, parenchymal brain invasion, which if you look in the clinical literature in the older times before we had effective acute lymphoblastic leukemia therapy, this was seen in children. It can occur. We don't know if it's happened in the children with this disease because they get so intensely treated, and if they relapse, they get treated so rapidly, and so now CAR T cells and other things, we're not sure if children actually have gotten this or not. But we decided to investigate the phenomena, and we did RNA-seq in our NSD2 wild type or mutant cells, and what we found, based upon what I've told you about what NSD2 does to activate genes, we found many more genes activated when NSD2 mutation is present across all 3 cell lines than genes being repressed. So when NSD2 is converted from mutant to wild type, we get a drop in gene expression. NSD2 supports a lot of genes in these mutant cells. And now we've done 4 different cell systems. We have 3 cells in which we remove the point mutation and convert it to wild type. These 2 B cells, and this is a T cell. And we have one other model, a T cell model, where we added back in this NSD2 mutation. So we can get sort of a consensus of, in this multi-dimensional Venn diagram, what genes are always activated by NSD2. And there are genes involved in TGF-beta signaling, hippo signaling, axon guidance, cell adhesion. So signaling and adhesion genes. In terms of signaling, we also found that there tends to be in a phospho, we did this in a kinase, global phosphokinase screen. We found that there's increased AKT signaling in these NSD2 mutant cells as stimulated by insulin here compared to wild type cells, and this is a quantification. So there's increased AKT signaling in these cells. And these NSD2 mutant cells seen in red are more sensitive to an AKT inhibitor than the isogenic wild type cells. So that might be a therapeutic hint. These cells have more AKT signaling. They might be treated maybe with these inhibitors. But this whole gene set we found in these cells, when we map it against what's called the Immunologic Genome Project database, these are genes expressed in, these 105 genes. They're not particularly highly expressed in B cells or T cells. They're expressed in stromal cells. Another way of looking at this gene expression, these 105 genes are mostly found like in fetal brain or amygdala. This is not a hematopoietic specific gene expression pattern. It is distinctly abnormal and turned on in these ALL cells. And it's relevant to patients because we did gene expression GSEA analysis, and we could find the same signature in human patients who have the NSD2 mutation in addition to other anomalies such as E2A PBX or ETV1 RUNX1. We also find that signature in mantle cell lymphoma patients who have the mutation, as well as to some extent in multiple myeloma patients. So this NSD2 overexpression or point mutation turns on this distinctly abnormal adhesion, neural invasion phenotype, because there's increased adhesion and gene expression changes. And there's some genes that are found all the time across some of our systems. And one of them is this gene that's called FGF13, but it's not really growth factor. It's an intracellular signaling molecule, and it has functional consequences in our ALL cell lines, because if we take an NSD2 wild type cell seen in blue, and this is the mutant cell, E1099K. Notice the growth difference. But here, if we take a wild type cell and add FGF13, we can recapitulate the 1099 increased growth. And by contrast, if you knock down FGF13 in an NSD2 mutant cell, we get this growth over here. And the difference seems to be FGF knockdown creates more apoptosis. FGF overexpression seems to decrease apoptosis. So FGF13, this is new data, could be one of these genes that's consistently turned on, that is a hallmark of an effector of the NSD2 effects on cells. This gene's turned on in every system we've looked at, in mantle cell myeloma, as well as acute lymphoblastic leukemia. Now, looking at this brain phenotype, what is actually causing this brain invasion phenotype? So one thing we actually asked about is what happens when leukemia cells are adhered to meningeal cells. Is there some kind of conditioning that occurs between the meningeal cells and the ALL cells to actually help them grow? So what we did is we co-cultured these acute lymphoblastic leukemia cells with meningeal cells, separated the cells, and performed RNA sequencing analysis. And what we found is if we take the NSD2 wild type cells and incubate them with blood-brain barrier cells or meningeal cells compared to this should be NSD2, I have this a little backwards. This should be a mutant versus control, and this should be wild type versus control. Anyway, the point of the matter is that NSD2 mutant cells stimulate, oh no, I have it right. NSD2 wild type cells with the blood-brain barrier cells turn on 752 genes. So just take an NSD2 wild type cell, put it against blood-brain barrier cells, 752 genes turn on. But in the mutant cell, only 151 genes turn on. So why so few genes get turned on in these mutant cells? The reason for it is that the pathways activated in an NSD2 wild type cell by meningeal cells are those hippo and extracellular pathway genes. And these genes are turned on by blood-brain barrier cells on an NSD2 wild type cell are already on in an NSD2 mutant cell. So it's a little roundabout way. Let me just say it again. Meningeal cells condition leukemia cells, turning on a neural adhesion pattern that maybe helps them invade the brain. But the NSD2 mutant cells already have it. So they've already got it. So it's something, this is a mutation selected for because it creates a more aggressive tumor. And this pattern of gene expression creates a more aggressive tumor. And it's similar to what's seen when you take blood-brain barrier cells and incubate them with leukemia cells. So what we could say so far is NSD2 turns on this aberrant program. It creates more sticky, invasive, adhering cells. They look like cells that are preconditioned to go to the brain, and in mice, at least, they go into the brain and create aggressive disease. FGF13 might be one of the genes that's involved in the proliferation, but I will tell you, when we've knocked it out, has no effect on invasion or migration. So look for the genes that are involved in invasion and migration. We've done a CRISPR functional screening, taking advantage of the fact that the 1099K cells go through a Boyden chamber more rapidly and are more invasive or migratory. And we could do a CRISPR screen in which we put in a library. We've done it with an epigenomic, a library of known NSD2 target genes, and we've done a genome-wide as well. And we collect cells from the top and bottom of the Boyden chamber. We quantify the number of guide RNAs, and we see which genes cause cells to migrate and what genes cause cells to not migrate, so we can determine this. And what we actually found is that there are some genes that when we knock out these NSD2 target genes, migrations does not occur, like this gene called PTPRG. It's a protein tyrosine phosphatase. And when we knock down PTPRG, knocking down its expression, we get decreased migration and we also have decreased adhesion. So this shows that we have a set of NSD2 target genes. We know that they're upregulated by RNA expression, and we do a functional screen against these, and we can identify some of these genes actually have a functional effect in creating this adhesive phenotype. And we like PTPRG because it's a protein phosphatase, and maybe it's potentially targetable. So it gives us clues to how to ameliorate these abnormal phenotypes. So what I've said to you this far is that NSD2 mutation increases leukemia cell growth in complex ways. It increases growth primarily in our ALL cell lines. It creates an anti-apoptotic signal, potentially through AKT signaling. I will tell you that that is particular to ALL because in mantle cell lymphoma, we see increased proliferation and S-phase entry. So these mutations may have context-dependent interactions. In ALL, it seems to be anti-apoptotic. Adhesion, migration is what's causing this increased leukemic growth. Additionally, NSD2 mutation turns on this abnormal gene expression pattern. Genes that should not be on in hematopoietic cells get turned on. We think these are functional. Genes like FGF13 can phenocopy the growth advantage phenotype. And FGF13 is an anti-apoptotic factor when we overexpress it. And there are genes that are involved with this neural invasion. And correlating this, we find that there are genes that are turned on by NSD2 that actually seem to drive a stickiness and invasion phenotype. So the eventual goal of this is to try to develop ways of therapeutically targeting ALL with NSD2 mutation, because the bad news is there's no NSD2 inhibitors as of yet. There's something that's been patented that we're trying to get a hold of to test it, but there's nothing in clinical trials. There are EZH2 inhibitors, there are other inhibitors, but there's nothing against NSD2. So we have to work around that problem. The last part of the talk, I'll say why is this NSD2 mutation, which I noted could be, could be in 1% of patients at diagnosis. Why is it found in every child cell at relapse? What is causing this relapse phenotype? Now, we did a screen with the National Institute of Health Center for Advanced Translation, and we took our isogenic NSD2 mutant and wild-type cells, and we screened these cells against thousands of anti-cancer compounds. And we found that most of these compounds were right on the diagonal line, no difference between the NSD2 high or NSD2 mutant cells, mutant or wild-type cells. But these compounds here were distinctly less effective in NSD2 mutant cells. There were a few compounds that seemed more effective in NSD2 wild-type cells, but they're more effective in NSD2 mutant cells. These are more effective in wild-type cells. These are more effective in mutant cells. These didn't really rescreen, but these rescreened very well. What were those compounds? These have they have a better effect in wild-type than mutant cells. These have better effect in mutant cells. These are all glucocorticoids. Every anti-leukemic drug, vincristine, daunorubicin, doxorubicin, L-asparaginase, 6-mercaptopurine, exactly the same efficacy in NSD2 wild-type or mutant cells. The only compound that doesn't work is glucocorticoids, a mainstay of every ALL treatment regimen, also mantle cell lymphoma, also multiple myeloma. It's essential for B-cell tumors. And here's the validation of this. This shows NSD2 wild-type cells responding to glucocorticoids. NSD2 mutant cells do not. This is in vivo. We inject mice with NSD2 mutant ALL, give them dexamethasone, no response. NSD2 wild-type cells, first of all, don't grow as well, and then they respond very well. And this is the extension of mouse lifespan with dexamethasone in NSD2 wild-type cells, but no effect in NSD2 mutant cells. So it's all about glucocorticoid. So what we find is NSD2 mutant cells seen in red given dexamethasone, no increase in apoptosis. NSD2 wild-type cells, the cells undergo apoptosis with glucocorticoids. That's how glucocorticoids work in acute lymphoblastic leukemia. They create pro-apoptotic cells. So then what we did to figure out what was going on, we did RNA sequencing analysis in NSD2 mutant cells and NSD2 wild-type cells. Take a look at this CEM cell line, which naturally does not have NSD2 mutation and is glucocorticoid responsive, a robust transcriptional response to dexamethasone. We add the mutation in, all of a sudden these cells become glucocorticoid insensitive and they don't transcriptionally respond. So why is that? And we overlap all these genes expressed in response to glucocorticoids only when NSD2 is absent. Common genes, these are all pro-apoptotic genes, and most notably, these effectors of glucocorticoids, BMF, BCL2, L11, also known as BIM, NF-kappa B, KLF13, all of these are only turned on in wild-type cells, but are not turned on in these mutant cells. So glucocorticoids can't work in these cells. And we did ChIP-seq for for glucocorticoid receptor in these cells, and we found the NSD2 mutant cells add dexamethasone. There's barely any signal across the genome. Remove the NSD2 mutation, now we get a robust binding of the glucocorticoid receptor across the genome. And here's an example of what that means. There's a critical enhancer in the BIM gene that turns on BIM in response to dexamethasone. In an NSD2 mutant cell, there's a very small peak seen here, but if we look at, but if we now take NSD2 wild type, we see that peak is already there with dexamethasone. Same thing in T-cell ALL. In fact, in T-cell ALL, there's no enhancer at all when you add dexamethasone when NSD2 is mutant, but when NSD2 is wild type, this enhancer that's bound by glucocorticoid is turned on. And what we also found is when NSD2 is mutant, there's a lot of K27 methylation at the BIM promoter. And when we lose NSD2, we lose that K27 methylation. And here is the dexamethasone-induced K27 acetylation at this BIM enhancer. This is present in wild type, but is below, it is absent in the mutant cells seen below here. So it turned out what we're saying, is there something wrong qualitatively with the glucocorticoid receptor? We said, well, maybe this NSD2 has got to interplay with the glucocorticoid receptor. It turned out to be much more simple. It turned out that NSD2 mutation inhibited the expression of the glucocorticoid receptor. You drove down glucocorticoid receptor levels so low that they were not able to bind to its targets and not able to turn on the whole glucocorticoid response. And it turns out that the glucocorticoid receptor drives transcription of itself in an autofeedback loop. But if you have too little glucocorticoid receptor, you can't even get that reaction started. So the cells are stuck at low glucocorticoid levels. You can't get genome-wide binding, and you don't turn on any targets at all. So this is true in our cell lines, and this is also true in PDX that we got from Richard Locke in Australia. And these PDX with NSD2 mutation have low glucocorticoid receptor levels. Okay, that's good. So I'm going to skip a little bit of this here and get to this major point here. So is this glucocorticoid receptor repression really the answer? And the answer is we could rescue these mutant cells by re-expressing the glucocorticoid receptor, and then we would gain back glucocorticoid response. It was as simple as that. We thought it was really a qualitative effect. NSD2 is doing something to the glucocorticoid receptor machinery, the way a SWI/SNF member might do, but it wasn't that. It was the levels were too low, and we can fully rescue by adding back glucocorticoid receptor to these cells. But you can't do that to a patient. You can't just give them glucocorticoid receptor. What could you do? Could you reactivate glucocorticoid receptor? We actually found that glucocorticoid receptor repression is one of only 3 genes that we found that are repressed by NSD2 in our ALL cell lines. There are like 200 genes up in every cell line, but only 3 genes that are down. And one of those 3 is the glucocorticoid receptor. And this was due to a pileup of EZH2, PRC2 at the glucocorticoid receptor promoter. No, why that is, I don't know. It's just really unique that there are some genes that are always upregulated by NSD2 and a few genes downregulated. And this turns out to be the glucocorticoid receptor. So we hypothesized if we treated these cells with an EZH2 inhibitor, and these EZH2 inhibitors decrease K27 trimethylation, this could resensitize cells to dexamethasone. And in fact, this happened. This is the vehicle-treated cells that are really pretty resistant to dexamethasone. But if we pre-treat with EZH2 inhibitor, now we can decrease cell viability with glucocorticoids. This was associated with increased apoptosis of these cells. So if we pre-treat the cells with EZH2 inhibitors and give dexamethasone, now you get increased glucocorticoid response and apoptosis. And this is also due to the fact that you can now induce BIM expression. This is BCL2L11 expression with dexamethasone. There is some induction in cells. It's a little less so here, but when you add the NSD2, the EZH2 inhibitor, you get even more induction here. And what we actually found by chromatin precipitation is that the NRC31 promoter, the NR3C1 encodes the glucocorticoid receptor. There's high levels of K27 methylation at this promoter, which drop when you give the EZH2 inhibitor. So we have less methylation at this promoter, and we now get the glucocorticoid receptor is now able to bind to the BIM promoter here, seen here, this is its own promoter. The glucocorticoid receptor binds to its own promoter. Seen here, very low level binding. It's not induced by DEX, but now when you give the EZH2 inhibitor, you get more binding of GR to its own promoter, and it increases with dexamethasone, meaning this gene can now auto-regulate itself, turn itself on. You get large levels of glucocorticoid receptor. It binds across the genome. It binds now to the BCL2L11 promoter seen here in this blue induction, and now you can turn on BIM more fully, and now you can kill cells. So this works in xenografts. If we treat ALL cells with a combination of GSK, which is EZH2 inhibitor, and dexamethasone, now we can extend the lifespan of these animals. So to summarize, NSD2 creates a lot of things in gene activation, but for reasons we don't completely understand, it represses like 3 genes, and one of the 3 genes is the glucocorticoid receptor. This likely explains why it's found in relapsed ALL. We recently had a meeting with the Children's Oncology Group to look at all these patients. The good news is that these patients are so aggressively retreated that most of them are doing fine because they get secondary therapies with other chemotherapy, CAR T cell therapies, but their early relapse is probably due to glucocorticoid resistance. We can reverse this, but potentially by giving an NSD2 inhibitor to liberate the glucocorticoid receptor from repression by NSD2, and now turn it back on, and we can cause a therapeutic response. So to conclude here, this mutation causes glucocorticoid resistance in ALL. It represses the glucocorticoid receptor, blocks its autoactivation. This low level of GR cannot bind across the genome. It doesn't do its job. But if you can reverse this by adding more GR to the cell, or by giving EZH2 inhibitor to allow the glucocorticoid receptor to turn itself on once again. And this what we've actually found this other agents that can further turn on the glucocorticoid receptor, which tends to be very frequently mutated. There are mono and biallelic mutations of the glucocorticoid receptor in ALL, as well as the related mineralocorticoid receptor mutation. So we found that DNA methyltransferase inhibitors and other G9A inhibitors seem to potentially reactivate whatever glucocorticoid receptor is left in the cell, turn it up more, and get more of a glucocorticoid response. So this may be a harbinger of potential ways to make glucocorticoids even more effective in acute lymphoblastic leukemia. And with that, I'd like to thank members of my laboratory, most notably Jianping Li, who just had this NSD2 mutation in ALL paper in Cancer Discovery, and Charlotte Kastner is a graduate student who's been doing those invasion adhesion assays and the functional testing. Manze Dalal has actually been focusing, she's a pediatric oncology now faculty member who's been working on FGF 13. We've also worked with Richard Locke's lab in Australia on these xenografts. Alex Mackerel did the modeling of NSD2. Neil Kelleher has done the mass spectrometry with us. Adolfo Ferrando, now head of research at Regeneron. I call him up one day. This is how science really works. This is how meetings like this work. I said, Adolfo, I don't understand why these cells are not responding to glucocorticoids. And he said, did you look at BIM levels? Did you look at GR levels? We did it. This is how collaboration works. He had the answer, and that's how we got this paper published, how it really happens. You can't know everything in your own head all the time. You reach out to trusted colleagues, you get some advice, you get a cancer discovery paper. Boom. So, but that's how it really works. So additionally, the National Center for Translational Medicine showed us that glucocorticoids were the only drugs that were different in these cells. There was other literature. We had some other things that maybe there were some other drugs. No, it was always glucocorticoid receptors. So just to indicate that it takes really a village of and collaboration interactions to get to the answer, and we couldn't do it without our funding sources. So again, thank you so much for being here. Those of you here, those of you online, and I'm happy to answer any questions.
Okay, questions? You know my style. I'll go for the PhD students first, and there are a few on that side, and then hand over to Alex.
Thank you for the presentation. It was really, really great. I enjoyed it really much.
I apologize, I tripped a little bit. I'm just, now the jet lag is kicking in, so some words, I'm having a little word finding difficulty, so thank you for being patient.
No, don't worry. I suppose it will be easy, the question. So, but I didn't understand. So NSD mutation avoids ECH1 to bind to the DNA, right? Is that the mechanism?
NSD2?
Yeah. NSD2 lowers the methylation 3 levels.
Absolutely. But it's interesting. It reshapes the epigenome. Yeah. There's increased K36 dimethylation that precludes K27 methylation at most places. But there are a few areas of the genome that it's like the polycomb EZH2 complex has to go somewhere. So it finds some safe harbor and it sticks there and it turns off those genes. We found that true in multiple myeloma as well. There are some genes that NSD2 indirectly turns off because it reshuffles the polycomb complex and goes somewhere where it normally doesn't go. And that's what happened in the case of NSD2 and ALL. It went to the glucocorticoid receptor.
That is why inhibitors work.
That's why the EZH2 inhibitors worked here, because in this case, it turned on the one gene that was really critical and repressed. Most genes are activated, causing that invasion migration phenotype. It's really weird, but stuff happens.
Okay, thanks. So this was Alberto Bueno from Manel Ester's lab. Can you also say your name and which lab you're from?
I'm Nacho from the same lab. Nice talk. And I'm wondering if you have studied the relationship between this mutation in NSD2 and the DNA damage response in order to analyze the effect on the tumor progression or maybe in the resistance of chemotherapeutic drugs? Thank you.
Thank you for that question. We have had some data on this in multiple myeloma. Steve Elledge did a genetic screen that actually found NSD2 as being important in DNA damage repair. It also was found independently that people did sort of a study to find all the proteins attracted to DNA damage sites in an unbiased manner. And one of the proteins attracted to DNA damage sites is NSD2. And what we found is NSD2 is attracted to DNA damage sites. And one of the things that NSD2 binds to a lot is PARP1. And we found that NSD2 helps recruit PARP1 to DNA damage sites and actually helps speed repair, in part, we believe, maybe not through its enzymatic activity all the time, but by ability to bind to PARP1 and help bring PARP1 to do its effects on many DNA damage pathways. In ALL, we don't have the data, but in myeloma, we have some data.
And have you analyzed the levels of H4K20 demethylation in presence or absence of this mutation in ALL or in multiple myeloma?
We have not. It's an interesting idea, but we have not done that. Thank you.
Thank you.
Okay, Sven? Ollie first, okay.
Hi, thank you very much for your talk. It was really cool. I have a question, and it might not be really well phrased, but you said that this mutation, well, one of my questions is you said the mutation was the most prevalent one. Do other mutations have similar effects? And then you said that it normally binds to areas of histone 3.3, but then there's a switch to 3.1. Have you chipped for this and seen like a shift into the gene body as well? Because as far as I'm aware, like, yeah.
Yeah, no, that was a theoretical calculation, and we have not experimentally shown that. We have not followed up on that. It's a good point. The T1150A mutation has never been studied in vivo. In vitro, it will cause increased catalytic turnover. Interestingly, one of our colleague Richard Locke's specimens with T1150A mutation was glucocorticoid sensitive. So is it possible that the T1150A creates some different chromatin environment than the E1099K? We're presently engineering that mutation into both acute lymphoblastic leukemia cells as well as mantle cell lymphoma cells, because the mutation is a little more common in mantle cell lymphoma than in acute lymphoblastic leukemia, to see if there's a functional difference between these things. And it could be because the surface area that's binding to the chromatin could be not only different in the catalytic turnover, but it could be somewhat different binding. So maybe we get this different activation. We're not sure yet, but it's an excellent question.
Can I ask one more little thing?
Okay, but only if you say your name, which lab you're from.
Oh, well, I'm from your lab, and we met earlier. And my other question was, you showed that you could recuperate normal chromatin environment with the H3K36 methyl 2 and the K27 methyl 3 by reintroducing a wild type or CRISPR editing. I can't remember what exactly you did, but you showed it as a Western blot. Did you ever chip for that and see that these kind of modifications were where they belong, or?
Yeah, so we've also done it by immunoblot. We've done it by mass spectrometry, and we can show that, again, there's about a threefold change. We can actually measure how many histone tails have a K36 dimethylation or a K27 trimethylation, and we can look at the exact number of those. And we could see that when NSD2 is overexpressed in myeloma, there's about an 8-fold increase in the K36 dimethylation. When it's point mutated, it's about a 3-fold increase. Now, in terms of the K36 dimethylation ChIP-seq, we had early success about 8 years ago with ChIP-seq, and those antibodies have gone. This is the dirty secret about us, about chromatin studies. There are some ChIP-seq antibodies that just are not as reliable as others. So our K36 dimethyl ChIP-seq, we kind of still see the broad pattern when NSD2 is a point mutated. I wouldn't be confident enough to say that there's, peaks and valleys that we can exactly map where. What we've seen, the very best we can tell, is that the NSD2 point mutation creates the mark everywhere, but I don't think the antibody is good enough to find peaks and valleys amongst that. So what we can find peaks and valleys of are the K27 acetylation. And what I think we might look at when we do the T1150A versus the E1099K is we're going to look at which enhancers get activated. Is there a difference between the two? But I don't think the K36 dimethyl ChIP-seq is going to be that informative. There's another question. Sorry.
Hello.
Yes, hi.
My name is Sven. I'm currently in the lab of Marcus, but I'm originally from the lab of Valeria Santini in Florence. And my question was, so this mutation in NSD2, does it affect the glucocorticoid receptor isoforms equally, or is that something you know or have looked into?
It affects the transcription from the predominant transcriptional start site. So the levels of protein differ because the transcription is differing. So whatever splice isoforms there are of the glucocorticoid receptor, I haven't checked that by immunoblot, but I would infer that all of them would be equally downregulated by NSD2 mutation because of that redistribution of PRC2 and EZH2 at the NR3C1 promoter. The whole gene expression goes down, so everything goes down.
Okay, thank you.
Sure. Alejandro, yes.
So great talk, I really enjoyed it a lot. So I have a couple of questions. The first one is, because you mentioned, right, and I think Alberto also touched the issue, that there is more life, beyond K27 trimethylation in the, K36 demethylation, the roles, that it has. So at which extent do you think part of the tumorigenic properties of this mutant of NSD2 are related to K27 trimethylation alterations versus K36, the role by itself of K36 dimethylation? So for instance, have you tried to look whether, besides the studies on relapse that you showed, whether decreasing the levels of EZH2, for instance, would somehow alleviate some of these properties?
Wait, I'm trying. Could you state that last sentence again?
So I was thinking is whether part of the phenotypic, the tumorigenic, all these properties, you have a wide range of things that you observe, right, that happen with these mutants. Whether part of it are actually the impact comes from an effect on K27 trimethylation. No, it's not by itself on altering dimethylation of K36.
Yeah, I think the K36, okay, so I think I see what you're saying. I am not super convinced that K36 dimethylation is interpreted as an activation mark. There are some, we talk about epigenetic writers and erasers and readers. There are some readers of the K36 dimethylation mark. For example, NSD2, its PWWP domain binds to the K36 dimethylation mark, so probably binds and binds a nucleosome that's K36 dimethylated, dimethylates it, then another molecule can bind to the next, one that's, so there's a self-propagation. But in terms of critical things like BRD4, which binds to K27 acetylation and sort of links enhancers and promoters and chromatin looping and brings in PTEF-B, very active interpretation of a K27 acetylation mark. I don't think K36 is the same way. So the way I interpret it is if you have K36 methylation abundant, you're keeping polycomb away, and it's the K27 trimethylation mark actively recruits repression because it brings in PRC1 and the ring complex, and that ends up bringing in chromatin compaction, and you compact chromatin. So K27 trimethylation is actively interpreted to repression. And if K36 is there instead, that K27 methylation is off the playing field, and therefore repression and chromatin compaction won't occur. And the K36 acetylation, which might be actively interpreted in chromatin looping, can occur. So I think that K36, we talked about, is it a sink? Is it sort of how a measuring metabolic states and activation states? Is it the K27 acetylation and the K27 methylation that may be the more active players, and K36 modulates those two? That's the way I'm thinking about it.
Very fast, very fast. So my only question, because maybe I misinterpreted, but so what you see is that this mutant of NSD2 doesn't really go to many different new places compared with the wild type NSD2. Is it the case?
Yeah, that I will say this. This one other thing. This is an important question. So we have not been happy with. We've made the NSD2 antibody that people use a lot as our monoclonal antibody. It's not been really good for ChIP-seq. So what we're doing now is we're epitope tagging with HA tag wild type and mutant NSD2 to get at that exact question. I have not had the tools to answer that question if the NSD2 is moving around to new places and doing new things. That is, I don't have the answer yet, but it's a critical question and we're answering it right now with the right tools. Yes, hi.
Yeah, hi. So I am Auralia Zanaska, working on adult TLL. So I have a couple of quick questions, but let's move to the patients now. If I catch it well, you mentioned that the frequency of NSD2 mutations in pediatric cases was around 1%.
I was saying, okay, the frequency of mutations in childhood ALL is about 15% of relapsed cases. It's also been found in about 15% of cases of mantle cell lymphoma. The 1% I was referring to was that the variant allele frequency in a diagnosis case of ALL tends to be low, meaning the mutation is present but at very low frequency at diagnosis and is present at very high allele frequency at relapse.
Okay.
But overall in patients, these are about anywhere from about 10 to 15% of cases, particularly in relapse cases.
Good, okay, because that was my first question, what happens on relapse cases? And my second question is, if you know if there is any association between this mutation in this gene and extramedullary invasion like central nervous system in patients in childhood.
Yeah, so the only thing I rely on is anecdotes. I know one of my friends who's a pediatric oncologist in the field, Kim Stegmeyer from Dana-Farber, told me an anecdote about a patient she had with this NSD2 mutation. She doesn't have archival mutations, archival tumors, but she told me this anecdote of this patient who had really bad brain and all kinds of things. I only have an anecdote. I've gone and spoken to the Children's Oncology Group to get that annotation. We just had a conference call. I just don't have annotation of this. And also the children are treated so aggressively, I'm not sure if they're getting to the point that they have all these complications, but so it's a little bit theoretical.
Okay, thank you.
Okay, two more questions, one from Fumi, and then Berta, and then we'll close.
First, thank you. I'm not working on ALL nor epigenetics, so my question is quite different. So this E1099K mutation in NSD2 gene is dominant in the gain of function. So that means this amino acid, glutamic acid, at this position is crucial and should be conserved. And how about the SNPs at this position in the human healthy individuals, and also conservation in other species of organisms? They should be conserved.
That amino acid is conserved across NSD2, and that is a conserved residue across species, and even the equivalent residue is pretty much the same in yeast. I'm pretty sure. There's only there's only one K36 methyltransferase in yeast, whereas there's multiple ones in humans, but that's pretty conserved. This mutation, I will also say, if you look it up and you go into the cancer dependency map and you look for this mutation, it is occasionally found in lung cancer, it's occasionally found in colon cancer, it's occasionally found in thyroid cancer. This mutation comes up rarely, but that point mutation comes up in some solid tumors, suggesting that this whole process could play again in other types of cancers. I hope I've answered that. Thank you. Oh, is it a normal? I don't think it's ever been seen as a SNP. There are indeed, you are absolutely correct in this question that there are some mutations in chromatin regulators that are found in congenital syndromes. The NSD2 deletion is found in Wolff-Hirschhorn syndrome, and they have a craniofacial malformation defect. And if you make mice that are deficient of NSD2, they recapitulate parts of the human thing. So the loss of NSD2 by intragenic, and there's actually a couple genes that are deleted in Wolff-Hirschhorn syndrome, including NSD2, creates a fetal malformation syndrome that is phenocopied by the NSD2 mutation it's not a mutation, but a deletion in mice. So, and many others, EZH2 and others can have fetal malformation syndromes as well as changes in human cancer.
Last question, Bertha.
Okay, hi. So very nice talk. So you said that NSD2 mutations were more frequent in infant leukemia, I think, or more abundant than the adult. And then my question is, do you think it might be related with other common translocations found there, like MLR rearrangements, that are so known to be, there's like a lot of epigenetic changes in those leukemias as well. So.
Right. So I understand the second question. The first question was, could you say what the first question was again?
Well, no, I think you said that NSD2 mutations were more abundant or more present in infant leukemia versus adult. I don't know. This is what I understood, but I don't know if that's correct.
So NSD2 mutations are found most commonly in mantle cell lymphoma, which is a intermediate differentiation B-cell tumor. It's found very rarely in multiple myeloma. There's one myeloma cell line in common use called MM1S that has the mutation, but no others do. It's found occasionally in chronic lymphocytic leukemia. In fact, want to hear a funny story? The way I figured that out is I Googled E1099K, and I found a graduate student thesis from Norway where NSD2 E1099K was mentioned in the PDF, and I found their thesis. So sometimes you don't know what to do with mutation. You Google it and you find something that was not even published yet. It was in a PhD thesis that hadn't been published. So it's found in CLL occasionally, and occasionally in solid tumors. Now, in ALL, it is particularly common in patients who have the E2A PBX chromosome translocation, and it's also particularly common amongst those who have what was used to be called TEL-AML1 or now called ETV6-RUNX1. It can be found in Philadelphia positive ALL. Currently, we are crossing mice with E2A PBX mutation to the 1099K knock-in mutation, which didn't have time to talk about today. The 1099K knock-in mutation by itself creates a B-cell, germinal center B-cell hyperproliferation, and upon serial transplant in animals, it can actually create a lymphoma-like tumor, but we're still interpreting that. So NSD2, that's not normal what it does. NSD2 mutations are almost never found by themselves. They're found in the context of other mutations. It's augmenting a phenotype. It's like making a leukemia cell look like it's touched the meningeal cells and ready to invade the brain. It's an add-on mutation. It's not the root cause, but it's a progression mutation. And that's how we have to understand it in terms of therapy resistance and more aggressive behavior.
Thank you.
Okay, thank you. For the benefit of time, I would say we stop here. Thank you again.
And thank you for these really fascinating questions that really stimulate more ideas for me. So thank you for the opportunity of being here today.